Thank you all for coming, and I thank the previous speakers. I'm looking forward to the next round of them. Let me tell you what I'd like to accomplish in the next 45 minutes or so. I want to define for you the trench to benchness of it all, or also known as reverse translation, and give you some examples. One example that we've worked for a long time on is prostate cancer. The other is radon, and it will give me a chance to give you some of the backstory to the work that Dr. Kim and our colleagues have been working on. And lastly, I want to mention some ongoing work in Parkinson's disease. And then I'll try and leave some time for discussing where I think this might go in terms of take-home message. We talked earlier about what translational research is. And again, the simplest definition, and one that I like, is that it's the process of turning observations in the lab, clinic, and community into interventions that positively affect people's health. So it's really pretty simple. You want to find something that's meaningful that you can apply that can improve people's health. Now, translation, there are a number of definitions of it in different parts of the literature. Sometimes people talk about T1 through T4, which I could never get straight. For me, the simplest way to think about translation is the direction that it moves in, okay? Now, the classic idea of translation is the so-called bench to bedside, the idea that somebody in her laboratory is going to develop some compound that they can administer to the public and the world will be a better place. The opposite end, looking through the telescope the other way, is trench to bench translation, the idea that you can actually find something in the population and then figure out what the cause is working downwards. This is the idea for much of epidemiology, or the goal. And the third type is sort of bench to trench, which might be conceived as implementation science, where you actually apply something to the community. So again, lots of things are easier to understand by example. I'd say the classic example of bench to bedside translation is Edward Jenner, who famously realized that because some people, mostly milkmaids, I guess it's the milk bench that got us to bench to bedside here, developed cowpox on their hands, did not develop smallpox. And this observation led him to collect pus from individuals with cowpox and scratch it under the surface of individuals in order to prevent smallpox. This was a bit of a controversial idea at the time, since many people thought that exposure to cow cells would turn them into cows. I believe this is the sort of evidence that the present Secretary of Health is concerned about with vaccinations, but no worries. This did not happen, and this was the basis for vaccination, which of course comes from vaca, for cow. Interestingly, Edward Jenner is now considered to be the founder, or the patron saint, if you will, of immunology, because this was at heart an immunological observation, although nobody understood the biology at the time. The classic and sort of amazing case to me as an epidemiologist of the reverse, trench to bench translation, has really got to be John Snow. John Snow was a physician in London in the 1840s and 1850s, at a time in which 1 million people in Europe died of cholera. Nobody knew what caused cholera. The idea was that it was maybe some bad air, miasma, some sort of undisclosed idea. This, of course, was before the advent of really understanding molecules as a cause of disease, the germ theory of disease. And Snow mapped the cases of cholera in London. and mapped them according to the provider, the water companies, and was able to show that what we would now call the epicenter of the cholera epidemic occurred in Broad Street, very close to a particular pump. Now, it turns out this pump was located 3 feet from a cesspit filled with human excrement, and the pump was pumping out fecally contaminated water. Again, nobody knew this, except when Snow made these detailed maps, he took them to the city leaders and said, there's something about Broad Street, there's something about this pump. Take off the handle of the pump. And when the handle of the pump was removed, the epidemic in London abated. And the idea of find the pump handle has become the mantra in epidemiology, the idea that if you can find the cause behind appearances, you can do something, and amazingly, you can do it without any knowledge of what the actual mechanism is or what caused the disease. Vibrio as a cause of cholera was identified by Robert Koch 20 years after John Snow died. But that kind of comparison, that kind of what is going on behind the facts is what reverse translation depends upon in many cases, and is the reason why John Snow is understandably considered to be the father of modern epidemiology. Somebody who thought very, very similarly was, of course, Sir Arthur Conan Doyle, who himself was a physician, and expressed many of his own ideas through the personage of Sherlock Holmes. And he said that in a study in scholarly, in solving a problem of this sort, and you might imagine this be a sort like what causes cholera, the grand thing is to be able to reason backwards. This is a very useful accomplishment and really an easy one, but people don't often do it. And when he expresses this to Watson, who of course famously doesn't know anything, and says, I confess I'm a little confused about this, he goes on to say through Holmes that most people, if you describe a train of events to them, will tell you what the result would be. That is, they could follow the sequence going forward. But there are a few people, if you told them a result, would be able to evolve from their own consciousness what the steps were which led up to that result. This power is what I mean when I talk of reasoning backwards. Now, sometimes reasoning backwards is not that hard. You could look at a result and probably put together, using the inner resources of your own consciousness, as it were, what sort of events led up to that. Other times, though, it's not so obvious what is the heavy, blunt object. And that's a lot of what reasoning backwards and a lot of what trench to bench is about. And what I want to talk about are 3 examples of trench to bench thinking and a couple of things that I've learned in my own experience from it. So I want to talk to you about prostate cancer. And if you're don't have a prostate, for example, or are not familiar with prostate cancer, the prostate is a 30-gram gland that surrounds the neck of the bladder. It functions directly to put prostatic fluid into semen and indirectly to put urologists into luxury automobiles. I say this because prostate cancer is the commonest cancer among men in the Western world. It's really, really common. In fact, if you were to look at the prostates of men who died from non-prostatic causes, they had a heart attack, they got hit by a car, whatever, by the time men are 50, at least 30% of us have microscopic prostate cancer in our prostates. By the time men are older, 70 or 80, we all do. So these microscopic lesions, which in most cases are clinically insignificant are extremely common. They're ubiquitous. And unfortunately, we can't really determine, looking from a biopsy, which lesions are important and which lesions could be left alone, which is what led to much of prostate cancer being overtreated. So subclinical prostate cancer is ubiquitous. However, what is not, and what is very, very different, this is a graph showing fatal prostate cancer. And the first thing you're going to see is that there is a 20-fold variation from fatal prostate cancer around the globe. Rates are very low in Hong Kong and Japan until, of course, these men migrate. And then within 2 generations, their rates triple. And rates are very high, highest in the world, actually, among non-whites in the United States and among Norwegians. Now, if you want to understand what causes fatal prostate cancer, not what causes latent disease, not what causes the ubiquitous form, but if you want to understand what causes fatal prostate cancer, then the question you have to ask yourself is, how did this happen? How are blacks like Norwegians? Because I'm going to go out on a limb a little here and say that superficially, blacks and Norwegians do not look alike. But they have to be alike. They have to be alike in some important way because they both have the world's highest rates for fatal prostate cancer. Blacks, Norwegians, and getting older because the rates go up dramatically with age. Now, again, if this were, say, Jeopardy, and you said, well, Alex, give me unusual diseases for 500, and they flash this picture up, and the music begins, dum, 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 dum. If you said, what is rickets? You would be right. Because rickets, as you know, is a disease of vitamin D deficiency. It's a bone-forming disease that became very common after the Industrial Revolution when the skies were filled with soot and sunlight, which is the source of vitamin D, was unavailable to many individuals. And the lack of sunlight causes bone-deforming disease, rickets. Well, when I got interested in this, I kept thinking, well, I don't know. But if lack of sunlight could cause rickets, I don't see why lack of sunlight couldn't cause another disease, which ultimately, by the way, is fatal because of bony disease, the kind of disease that President Biden has. So not knowing anything, but trying to think backwards, we wrote a paper, this is quite some time ago, arguing that maybe vitamin D deficiency underlies the major risk factors, being old, being black, and living at northern latitudes. And in order to see how that would work, let me just remind you very briefly how vitamin D is synthesized. And I say synthesized because you call it a vitamin, but it is not. It is not. Vitamins, you probably remember from high school biology, are things that your body can't make and must ingest from outside sources. Despite its name, vitamin D is not a vitamin, it's a steroid hormone, and it works like this. Sunlight acts on a precursor in the skin, 7-dehydrocholesterol, and it turns it into vitamin D. Vitamin D is inert. You can also eat it if you want, but 90% of the vitamin D in your body comes from sunlight. The liver converts vitamin D into 25-hydroxyvitamin D. It puts an hydroxy group at the 25th carbon. This is the major circulating form of vitamin D, but it is inert. The kidney turns 25 into 125. 125-dihydroxyvitamin D is the active steroid hormone. So that's all you need to know about vitamin D. If you want to remember the synthesis of it, remember the word silk. Skin or integument, SI, liver, LK, kidney. That's how vitamin D synthesis works. Well, how could that help us understand the risk factors for prostate cancer? Well, if you take a light-skinned individual and a dark-skinned individual and expose them to the same amount of ultraviolet light, it will take the dark-skinned individual 10 times longer to make the same amount of vitamin D as the light-skinned individual. And not surprisingly, in any population-based survey like NHANES and others, you can see that vitamin D deficiency is 10 times more common among African Americans than among whites. Well, this idea starts to get traction when you realize, well, what other predictions does it make? And these are two maps. These are called trend surface analyses, which medical geographers will explain, give you a three-dimensional map of some process. So this was before advanced computing became available in the 90s, where we actually took values of ultraviolet light, of albedo, cloud cover, and calculated the amount of ultraviolet radiation that hit every one of the 3,073 counties in the US. And this took some time. And you will probably not be shocked to realize that there is more sunlight in Miami than there is in Minot. Okay, so this was really not the news portion of the broadcast. But if you look in whites, and these data are all age adjusted, and ask, what is the best fit relationship between geography and mortality? Again, fatal cancer, not incident cancer. Well, the surprising finding is that mortality rates from prostate cancer, when the effects of age are pulled out, are in fact quite significantly higher the farther you go north, and in fact are very significantly associated with the inverse of the availability of ultraviolet light. Now, when we had published the first idea a couple of years ago, we were lucky even to get it out. It was very difficult to get it published. When we finally got this published, and it ran in the New York Times and in the Chicago Sun-Times and many other places, the idea that sunlight, the commonest exposure you might have, was somehow related to prostate cancer, the commonest cancer in men, suddenly got greater traction. And we were able to actually start a series of laboratory-based studies. And I'm going to summarize more than a decade of work in a single slide. But I would point out that what we found, and my colleague Gary Miller showed first, that prostate cells have receptors, 1,25-dihydroxyvitamin D receptors in the prostate. This was not surprising because we had found it in the mouse and actually had published that earlier. But if you expose prostate cancer cells in vitro or in vivo to vitamin D exerts profound anti-cancer effects. So the hormonal form of vitamin D inhibits the proliferation, the invasion, the metastasis of prostate cells. And to give you an example from one of our early experiments, these are lungs from 3 groups of rats given a rodent form of prostate cancer. These are rats given prostate cancer and no vitamin D hormone. These are animals given the vitamin D hormone and cancer. And these are animals' lungs from rats given the cancer and an analog of the vitamin D hormone. And all this lumpy, bumpy stuff, which looks like peanut brittle, that's tumor. So you don't need to be an epidemiologist although it helps. But you really don't even need to be an epidemiologist. You can look at this grossly and see that 125, the hormonal form of vitamin D, has profound anti-cancer effects in the laboratory. And then, of course, we began to study men who had high solar exposure. And in large prospective studies, most of the evidence shows that men with high serum vitamin D, high serum levels of 25-hydroxyvitamin D, which you'll remember is the major circulating metabolite, have a lower risk of aggressive or fatal prostate cancer. So this was super exciting. And as I say, it's over decades worth of research. But there was a little bitty problem. The problem is this. This is the slide you saw before. And yes, vitamin D synthesis is skin, essentially. Sunlight or integument, liver, and kidney. Does sun produce vitamin D? You betcha. Does the liver convert vitamin D into 25-hydroxyvitamin D? Yes, in an unregulated fashion. So the more sunlight you have, the more levels of 25-hydroxyvitamin D. The levels of which can be quite astronomical, for example, in lifeguards at the end of summer, because they're always exposed to sunlight. Is 25 lower in blacks and at high latitudes? No question, absolutely. The problem is 25-hydroxyvitamin D is inert. And the kidney converts 25 into 125, the active hormone. The active hormone, 125, is not lower in blacks than whites and is not lower at higher latitudes. So we almost got there, except that we failed in the last step. How come this doesn't work? It should work. This kept me up more nights than I can possibly tell you. Why doesn't this work? What is the Do I not believe that north-south gradient based on 3007? Yes, I do. Do I not believe what everyone has been able to show in the laboratory, including ourselves? Well, it's hard not to believe that because everyone's shown it. Where's the screw up? And then, I don't know what it was, but I finally realized, oh, there is a solution to this. The solution to this would occur, and again, reverse translation. How could this have happened? The solution to this could occur if the prostate gland had 1-alpha hydroxylase, if the prostate could make its own hormone. The problem with that idea is that every textbook said that was impossible until we showed it to be true. And for those of you who are laboratory people, I'll briefly tell you that we showed it to be true by going into the operating room, getting prostate cells at prostatectomy, growing those cells up in primary culture, and adding radioactive 25-hydroxyvitamin D. And then assaying the cultures for radioactive 125-hydroxyvitamin D. So if there's any radioactive 125-hydroxyvitamin D in those cultures, there's only one way it could have happened. The prostate cells put it on. And we did it that way because the gene for 1-alpha hydroxylase had not been cloned. 6 months later, the gene was cloned, and everybody showed it to be true. Every prostate cell examined has 1-alpha hydroxylase. In fact, the prostate, the humble little prostate, makes more active vitamin D than the kidney. It's just that it's not released into the circulation. So what we knew before is, sunlight adds some vitamin D becomes 25 in the liver, 125 in the kidney. Does that inhibit prostate tumor growth? Yes. But what we learned from this was that the prostate has the enzyme that turns the water into wine. So you don't need a kidney, at least as far as your prostate is concerned. And the reason that that's important is that that provides a mechanism by which sunlight could actually inhibit fatal prostate cancer. So what was very gratifying to us is that, at least in this system, we were in fact able to go from the level of geography to the level of the gene and discover a mechanism that is key to prostate cancer prevention. And believe me, there are many men like myself who take vitamin D because it's a very effective way to maintain the differentiated phenotype of prostate cells. Excuse me. However, in the present context, what this sort of changed my whole way of thinking is that it made me realize that this basic approach, reverse translation, can be applied to lots of conditions. Okay? So yes, we learned something important about the prostate, and I really enjoyed it. However, I'm convinced that the idea of going from present observations to try and think backwards, what caused this, is a terrific research method. And I want to give you some examples, the backstory for some of the excellent work that my colleague, Dr. Kim, in our group has been doing about radon. And basically, all you need to know about radon is in this slide. Radon, as I'm sure you know, is a radioactive gas that comes from the decay of uranium in soil. It accumulates in homes because it's under pressure, it gets into your home, and because where it's cold, in particular, you sealed up your home, so the radon gas gets in, but it doesn't get out. And this is particularly problematic in North Dakota because North Dakota has the highest radon levels in the United States. As a rough estimate, levels of radon in Grand Forks are 9 times greater than the US average. And there are some homes that are the equivalent of smoking 2 or 3 packs of cigarettes a day. So radon is a huge problem. And we were thinking, just to give you an idea how big a problem it is, you probably understand that radon is a problem because it undergoes radioactive decay. Radon itself is inert, it's an inert gas. However, it's in the process of becoming polonium, and then polonium becomes lead. And as it becomes polonium, it liberates energy, and that energy damages DNA. So more people die every year from radon-induced lung cancer than die from drunk driving. But you don't hear people talking about radon. And in fact, there are diseases like stroke, which is even more common than lung cancer, which our group has recently shown are likely caused by exposure to radon. So radon is a huge public health problem. And my worry about this is, how come nobody talks about it? People don't know much about radon. And maybe it's because, yeah, sure, it's invisible. But maybe people don't know anything about radon because it's our fault. Maybe because our communication about radon is so ineffective, which is why Sujung and I have had the pleasure of working together. Think about it. How do we actually communicate about radon? Well, largely through the print media. We hand out brochures from the EPA describing the hazards of radon. Well, if you had an important message you wanted to convey to somebody, like, hey, dude, your house is radioactive, would you print out a brochure and stand out in front of Target and hand that out? Or would you email that to 2,000 of your closest friends? So we reasoned that, you know what? Maybe we're at fault. Maybe we're the problem behind the appearance. So we took the information in the EPA brochures, didn't change it at all, except we turned it into an app for phones. And then asked, well, would people respond differently to a brochure than their phones? I'm not the sharpest knife in the drawer, but I see people using their phones a lot. And I'm thinking, well, what if we took the same information, turned it into a phone app, and then compared it head to head? Okay? And I say this in the context of TRANSCEND is that the NIH fully recognizes that the most definitive answer to any kind of question is a randomized or controlled trial in people. So why not expose people to the radon information via their phones or via the standard procedure? Ask how much do they learn about radon? And importantly, how likely are they to order a free radon test kit? So basically, this is the standard consort diagram, which we've done now 3 or 4 of these trials, and the results are pretty much the same. But take individuals, exclude individuals who've tested previously, have 100+ individuals, randomize them, to the radon app or to the brochure, expose them to information about radon before and after to see what they know, and then also see how likely they are to order a test kit, so that the individuals who received a brochure had a prepaid envelope or prepaid card, said, just return this and we'll send you a free radon test. The individuals exposed to the app said, just press here and we'll send you a free radon test. And this pre-post, pre-post, pre-post is essentially a measurement of how much they know about radon, what their attitudes were, other items in the health belief model that Dr. Kim mentioned. The important thing to note is that in the pre-exposure, these are true-false questions. These are not complicated questions. These are questions like, radon is a gas, true or false? Radon is invisible, true or false? Radon causes cancer, true or false? And what you see in the pre-measurement is that these are true or false questions, and people's response to this is chance. Okay? This was done in Grand Forks, North Dakota, the most radioactive county in the most radioactive state. And people don't know the difference between radon and Ray-Bans. They will learn about radon through the brochures, and they will learn about radon through the app, just that they learn better using the app. And in fact, the acid test is, how likely are they to order a test kit? They are 3 times more likely to order a test kit through the app. Now, Sujung is now working on an interesting question. Yeah, people order the app, but they don't often use the test that they get. Yeah, that's another matter. That's like cutting off the Hydra's head. You think you solved a problem, but you haven't. You've just created 2 new problems. However, those problems are solvable, and we're working on solving them now. So the bottom line from this intervention is the app superior to brochures? Absolutely. It's better on radon knowledge, it's better in self-efficacy, and it's way better on requests for radon tests. So here's an example in which the problem is not what caused this guy to be dead on the floor or what causes cancer in different populations, but what causes a lack of knowledge and how can we fix it? The app is one way to fix it. And many times there are untold consequences of this. We presented some of this information at a radon conference, and of all places, the International Atomic Energy Commission wanted to know if we could translate this into different languages and use it in Europe, because it is a fact that the largest exposure of any individual to ionizing radiation is from radon at home. So if you want to limit the carcinogenic effects of radon, you need some way to communicate them better. And this is a very simple way in which an intervention at the level of the population leads to an intervention that changes people's health. Now, the last example I want to talk about is work that's ongoing. But first, the summary here, of course, do smartphones communicate radon risk? Yes, very effectively. In randomized trials, and again, we've done 3 of them now, they are much better than brochures in increasing radon knowledge and increasing test requests. I do believe, and was shocked to learn really, that smartphones could become the standard intervention to increase radon knowledge and testing. And we're actually in negotiations with the state health department because we're working to fix that app so that people actually use the tests and then just give it to the health departments. So the last example I want to talk about, and maybe leave a little time for discussion, has to do with Parkinson's disease. This is of interest to me because, as Kent mentioned, before I was an epidemiologist, I was a psychologist, and Parkinson's disease, as you probably know, is the most prevalent neurodegenerative disease. About 2% of Americans over the age of 60 have Parkinson's. it is a very, very important cause of loss of quality of life. There is sadly no known means to prevent Parkinson, and presently no therapy that modifies the actual natural history of it once it begins. So that, yes, there are therapies for it, but it does not change the natural history of it. And the odd thing about it, I think the really odd thing, is that we have a very, very sophisticated body of evidence about what the pathophysiology is. Sometimes people say, what's the cause of something? And they mean, what's the pathophysiology? It's not the kind of cause I'm talking about. I'm talking about what is the heavy, blunt object, not the pathophysiology of concussion. So we know that persons with Parkinson's lose cells in the substantia nigra, which is just Latin for black substance, the part of the brain that makes dopamine, and that neurons that are affected by Parkinson's are less able to uptake dopamine at the synapse. And I have to say that it's an embarrassment to me almost as an epidemiologist that we haven't figured this out. It's got all the ingredients that suggest it should be figure-outable, which maybe if it's not a word, it ought to be. Because there's a lot of it. We understand the pathophysiology. And it's not distributed randomly. And this is, to me, one of the most interesting things. There is a belt of high incidence of Parkinson's disease that is pretty much stretches from the northeast to the Midwest. There's some in Texas and some in California, but there's a Parkinson's disease belt. And these data are quite good because they're based on pharmacoepidemiologic data, which is just a fancy term for tracking drug prescriptions. If you have Parkinson's, you are almost assuredly on L-DOPA replacement therapy. That's covered by Medicare. So we can track how much Parkinson's there is based on how much the prescription usage for the replacement therapy. So now we're in a slightly more challenging situation, is that what's that about? Okay. And again, it's a little bit more challenging than the guy dead on the floor. And the trick is to, and I only wish that Conan Doyle was a little bit more explicit about evolving from your inner consciousness what the events would be that led up to it. I think that's just a very ornate way of saying, come up with a reasonable hypothesis. One reasonable hypothesis would be that whatever causes this has a distribution similar to it. Sort of saying that you need to be at the scene of the crime in order to be a suspect. So we tried a number of things, and one of the things that really interested me is there is a belt that stretches from the East Coast to the central US, and it roughly follows industry. And one of the problems with industry is that it releases pyrolysis products into the air, which acidifies rain. So acid rain shows a very similar distribution. And what we did is we calculated how much acid rain falls on all states by simply taking the pH of the rain and multiplying it by rainfall. And it's not perfect, but there's a pretty good relationship between the acid rain index and the prevalence of Parkinson's disease. And then the question is, again, using your inner consciousness, as it were, how could that be related, if so? Well, actually, one of the things about acid rain is that it dramatically increases the amount of metals in the environment. It causes some statues, in fact, to disintegrate because the acid rain mobilizes metals in water. Water containing lead turns into blood containing lead, and blood containing lead winds up in the brain. And maybe there's some way that metals in water could contribute to Parkinson's disease. And if that were true, we actually dug up, after some time, You know the pipes that lead to your house carrying water? They're mostly lead. They're called service lines. And if you calculate the logarithm of lead service lines and the logarithm of Parkinson's disease prevalence, there's a surprisingly good relationship there. All right, well, what do we need now? How do we move this into something else? What you need now is to make friends with some laboratory scientists, like my friend Jamie Foster. And what Jamie has been able to do with his group, and I don't want to steal too much of their thunder because they have a poster, and I strongly recommend that you go check it out during this afternoon, is that they've exposed rats to lead in water, comparing lead acetate to sodium acetate as the control at different doses, and then decapitated the rats, taken out their heads, and not like Hercules decapitated them, but in his own way, maybe he is, and looked for the effects of lead on synaptosomes in the brain. And the rats exposed to lead in drinking water are less able to take up dopamine, which is one of the symptoms that, or signs really, that you find in Parkinson's disease. So the idea here is that maybe you really can go from trench to bench. Maybe you can actually start with something and maybe make a couple of guesses or evolve from your inner consciousness what the events were that led to that result. And that's what reverse or back translation is. So what's the take-home message? Trench to bench, the idea that you could start with findings in the population and work downwards towards the lab, it's the real deal. Is it a legitimate form of translation? And it sometimes produces unusual results which would never have been found otherwise. Trench to bench is especially useful in diseases of unknown etiology, and amazingly can actually protect public health and save lives even when the disease and or the mechanism is completely unknown. Johnson is a perfect example of that. If you think in modern terms, there are other examples. Think about sudden infant death syndrome. We don't know what the mechanism that causes babies to die in their cribs. But we do know that if you put babies to bed on their backs, they are much less likely to die, which led to a public health campaign called Back to Sleep. Trench to bench thrives in environments where there are both population and laboratory scientists. If you're a lab scientist, take an epidemiologist for coffee, and the reverse. Promoting those sorts of interactions is one of the goals of TRANSCEND, and it's been a pleasure to work on them. I'd like to thank the National Institute for General Medical Sciences for the TRANSCEND grant. Our work on radon has been supported by a number of agencies, and I really want to give a very big thank you to our many collaborators at UND and to the dedicated folks working on TRANSCEND to make the symposium and to make this work possible. Thank you for coming and for your attention. I think we've got a couple of minutes before lunch, so if you have questions, I'd be happy. Yes? Ashley? Loud, loud. Maybe we'll get you a microphone. Yeah, thanks for that question. It's a very good one. There are two forms of vitamin D that you can ingest, ergocalciferol and cholecalciferol. Ergocalciferol is a vitamin D2. It's from plants. Cholecalciferol comes from fatty fish. in pretty much every laboratory assay, ergocalciferol and cholecalciferol are equivalent. I would say that vitamin D3, cholecalciferol, binds slightly better to the 1,25-dihydroxyvitamin D receptor and might be preferred. But in human studies in which the distinction is made analytically between ergo and chole, first of all, most of it actually is vitamin D3, but there's no difference between vitamin D2 and vitamin D3. So wherever the source of that matters less. As I say, the majority of individuals get their vitamin D from sunlight. There are very few sources of so-called vitamin D in foods other than fatty fish and shiitake mushrooms.
Thank you. That was a fascinating talk. I have a question also on the first part of the talk.
Yes.
Right here. So, is it, to go full mechanistic, is it known why prostate cells need to make their own vitamin D? What's the physiological process that these cells use vitamin D for?
We used to think that the kidney was the only source of vitamin D, and all the textbooks in the 1980s insisted that only the kidney made 1,25-dihydroxyvitamin D, and the evidence for that is a little imperfect in terms of reasoning. The evidence was that individuals who are anephric, who had no kidney, have no circulating 1,25-dihydroxyvitamin D in the circulation. That's true. However, that does not mean that other cells do not manufacture their own 125 for their own purposes. So if Arnold Bread and Wonder Bread suddenly stopped making and selling bread, and no bread was available in grocery stores, that doesn't mean that people don't eat bread. It just means that they're making their own. It turns out that 125-dihydroxyvitamin D, in addition to its role in calcium uptake, plays a major role in maintaining the differentiated phenotype of cells. So while people were shocked that we showed that prostate cells have 1-alpha hydroxylase, we later went on to show that the pancreas and other cells often have it as well. And it's now understood that 1-alpha hydroxylase is ubiquitously expressed by many cells that manufacture their own 125, and 125 in virtually all of those circumstances helps maintain the differentiated phenotype of cells. So very surprising finding at the time. The difference is that 125 is very important as an endocrine hormone from the standpoint of the kidney, but it is an autocrine hormone. And autocrine means you just make your own in many other cells. So skin cells, pancreatic cells, prostate cells, breast cancer cells, most cells actually, of which I am aware, actually have 1-alpha hydroxylase. And that's one of the reasons behind the widespread idea that vitamin D supplementation is probably a good thing for cancer, because it will maintain levels high enough that cells can manufacture their own 125 that has a role in maintaining a differentiated phenotype. Matt.
Thank you. That was a really compelling presentation of going from bench to trench and trench to bench. And the examples were wonderful. And I'm wondering if you have a chance to drill into the brain the way you drilled into the prostate for sort of final arbiters of the mechanism of disease. Is there higher lead in the brains of patients with Parkinson's disease than those without? And a related question is, do people who have, say, evidence of lead poisoning, like kids in early life, have a higher incidence of Parkinson's disease later on?
Great questions. Thank you, Matt. The first thing I want to point out is that the first clue, obviously everybody I'm sure knows that lead is a neurotoxin, which is why lead paint is so dangerous, because it produces retardation in children. And there's a very good relationship between lead levels in children and IQ. But the first clue that Parkinson might be related to lead was a case report. 3 individuals working in a lead battery factory in Germany. And these individuals developed Parkinson's disease, and their work exposure was later described. And this was in 1967. There have been a number of studies, subsequent studies, showing that lead exposure contributes to Parkinson's disease. Lead is a little tricky to measure because blood lead reflects recent exposure, not lifetime exposure. However, bone lead reflects lifetime exposure. Work done by my colleague, Dr. Weisskopf at Harvard, has shown that bone lead predicts Parkinson's disease. And the data are mixed, but do individuals with Parkinson's often have elevated levels of lead and a couple of other metals? Absolutely. Absolutely. It's not 100% clear, and part of the problem is has to do with the half-lives of these chemicals and how long people were exposed to it. But to me, the most definitive thing was those initial case reports of the 3 lead-exposed battery workers.
Thank you. A follow-up question is sort of your grand vision of linking a trench and the bench. Do you have any notion of how to set up, say, informatics systems in the CTR and the university and combine it with surveillance systems to sort of have the virtuous cycle of translational research from out in the field to in the university where you have the scientists looking at basic mechanisms?
That would be my dream. I would love that. I would love that because it's hard to read somebody like Sir Arthur Conan Doyle because he's just so amazingly knowledgeable about all sorts of things. And the idea that anyone should be able to draw upon her or his own inner consciousness to come up with this is hard to fathom. However, with something like AI, we should be able to understand what are the patterns shown by any number of these things, and show me 100 patterns. Do the work for me, please. It would be wonderful because then you would be able to plug in an answer and say, what does this look like? And a computer would either say, oh, well, this is rickets. Well, no, it's not. Or it's prostate cancer. Or it's some nephrological condition. But it would be wonderful if we could come up with a way to do that because it would save so much work. And it would also save so much criticism because it's not hard to publish papers that are hypothesis testing. It is very hard to publish papers, unless you're a senior person in the field, arguing that this is what I think makes sense, and this is how it would work, even if you had data. So that the paper that we showed showing the geographic determinants of prostate cancer mortality being inverse of sunlight has now been cited over 1,000 times, but it was in review for an entire year. It was the worst year of my life. We sent it to Cancer, it got rejected, and then the editor wrote back to me saying, look, Gary, I think you got a bad deal here. Can you make these changes and send it back to me? That happened 4 times. Because who was I? I was a postdoc. I might have better luck now, I'm not entirely sure. But it's very easy to be seen as an hypothesis tester because that's what everybody understands science to be. There's this weird sense of, oh, where hypotheses come from, I don't know. I can't, yes, they're extra scientific. They are extra scientific. They're not part of the normal corpus of science, and it is very difficult to publish those things. And if you could actually put the onus on a computer, that would be great. You could say, well, this isn't me, but I got this output, and therefore I'm testing it. It would save a lot of problems.
Gary, very nice talk. I particularly liked the way you were trying to explore the segments of the bench to bedside model. And given that, I'm wondering what you think about the National Center for the Advancement of Translational Sciences notion of the way to really bring this to patient care is to get multidisciplinary groups sitting at the table where you have basic scientists, translational scientists, clinicians, implementation scientists, data managers, data scientists, communications experts. What do you think about integrating people in that way to try and bring this to fruition? And how are we doing with that in North Dakota?
Well, what you described would be ideal in many ways. And I think the first element in that, which is why I strongly recommend you take your badge and come up to somebody and say, hey, what do you do? Here's what I do, is that you can't collaborate with people you don't know. It does really take a tremendous number of people with different skills to actually execute on one of these things. I remember very vividly being at a conference where I asked the world's expert on vitamin D synthesis, Michael Hollick from Boston University, and I said, look, what do you think about the idea that the prostate has 1-alpha hydroxylase? He said, oh, it's ridiculous. I said, yes, but other than that, other than that, if I could provide you with prostate cells from a recent operation, would you want to work with me on that? He said, oh, sure. And of course, when the paper came out, Mike explained how we had hypothesized this for years. Fine, no problem. I think everybody deserves credit for working on these things, but if you don't have the right people, then you get to a certain point and you just stop. And I must say that while I find a lot of the jargon on some of the NIH websites as kind of really lofty, the National Center for Advancing Translational Sciences actually has some text that I can really relate to. You may know that the CTRs, like the TRANSCEND study we have, is a kind of local version of the translational grants given by a larger branch of NIH. And there are more established, better muscled, more experienced, but not necessarily smarter universities that have the parent grant of these. And these are $40 or $50 million enterprises. And some very interesting things, I think, will come out of that. But I am a big fan of the goals of the National Center for Advancing Translational Science. We're not as far as we want to be, but I see this as a first step. And you are just steps away from lunch. So yeah, get some lunch, get a friend, find somebody you don't know, sit next to her or him.
All right, let's give another round of applause.